Lovely.
Hi.
I'm going to go with you're not actually there, just like I'm not here.
Well, I'm kind of around the corner. We're both in Cornwall.
Oh, you're always in a cool place, actually. Um, so I want to introduce Linda Partridge. Linda is coming in from actually a couple of places, but we'll say University College in London. And she has been a pioneer in the use of flies to study aging, and in particular, uh, thinking about the therapeutics and how those can affect, how those can be used to begin to manipulate aging. So Linda, please take over.
Thanks very much, Ross, for inviting me. And hi, everyone. It's been a great series of talks and also really lively discussion afterwards. It's terrific. So now for something completely different, I'm going to talk about aging. So as Ross has said, I'm based at UCL. I also have a lab in Germany at the Max Planck Institute there. Both of them involved in work on biology of aging, and especially in ways that we can intervene in the aging process to try to improve health. So that's really what I'm going to talk about this afternoon. And I'll get my PowerPoint, and yes, it's there, so I'll share the screen with you. Can I do that from here? Yes, I can. Okay, so here we go.
That works.
So the basic theme, intervening in aging to prevent aging-related diseases. And I think looking back through the talks that you've had, that you probably haven't heard much about aging. And, you know, I don't know how much you've about it outside the proceedings here. So I'll just do a bit of a general introduction and then move on to what we can do with flies, because I think they're a great organism for working on aging. So why is it important? You know, why bother to study aging at all? It might seem a bit off stage left. And the real reason is that it's an increasing global challenge really for countries worldwide. And that's because the global population is aging. So these are data from the United Nations published last year, where they've simply looked by year at the representation of different age classes of humans in the world population. And you can see what's happening. So there was a peak in young people in the kind of 1960s. These are people aged 5 to 14 and also newborns in the small orange line at the bottom. But since then, that proportion's declined steadily, as has the proportion of 15 to 64-year-olds actually. And what we're seeing is this very steady increase projected to go on now till the turn of this century in people aged 65 and over. And that's driven partly by birth rates. It's not entirely an aging thing, but nonetheless, we've got this increasing proportion. And it's reflected in the fact, and partly contributed to by the fact that we're seeing this big increase in human life expectancy. So again, this is a paper from some years ago now, but I could have shown you a lot of different graphs of this, but I think this is a particularly telling one. And basically this trend has continued. So this is a couple of demographers, and what they've plotted is life expectancy in years up the y-axis against the year in which the birth took place for the country that was the world leader at the time. So this tended to be the Antipodean countries early on. Latterly, it's actually been the Far East. At the moment, South Korea, I think, is the world leader for both sexes. And what you can see is this extraordinary 2.5 years per decade, 6 hours per day, if you prefer, increase in human life expectancy. And as I say, this trend is happening worldwide. At the moment, it's happening most rapidly in middle-income countries. The more developed countries are slowing down a bit, but the trend is tending to occur everywhere. So, I mean, what we've got here is in many ways something to be celebrated. People have got better at looking after themselves. They're staying healthier for longer, and so they're living longer. The reasons for the trend's been different at different stages. There's been clean water, better food supply, antibiotics, immunization, very recently better healthcare of the elderly. They've all fed in at different points in the trend. But what's happening is that we're now living much, much longer than we did in our evolutionary past, where most people would have been dead certainly by the age of 40 and probably sooner. We're seeing these massive increases in lifespan past the point at which, you know, natural selection would have been able to look at them and sort out performance at different ages in the past. So we're way out of, you know, equilibrium with our evolutionary history. And although the trend is something to be celebrated, it's coming with a very obvious downside, which is this one. So this is from the Global Burden of Diseases, published about 3 years ago. And what we've got is 2 low income on the left, 2 middle income and 2 high income countries by world standards. And the incidence of cardiovascular disease, dementias and cancers at the bottom. And what you can see is this very steady increase. I mean, that's the big thing across the picture. The elevation of the lines is different. I mean, we've got very different diagnosis rates for these diseases in different countries. So what we've done here to try and partly control for that is to normalize everything either to the incidence at age 20 for cardiovascular disease and cancer, or to age 40 for dementia, which tends to be a later onset condition than the other 2. But even so, you can see, for instance, for cardiovascular disease, it's very much more likely to be diagnosed in the high income than the middle income than in the low income countries. And sorry, for cancer. And also for cancer, you can see the big sex difference in the incidence. So the pale sort of beige lines here are for women and the green lines for men. So what we've got is this increasing lifespan. We see this increase in age-related diseases. Not just that, but there's frailty, loss of function of various kinds, just general old age impairments that fall short of disease, but which of course cause a lot of human misery. And also these age-related diseases are particularly in the higher income countries, now giving rise to something that's become known as multimorbidity. So what you find is patients turning up in hospital or at their general practitioners who've got several things the matter with them, not just one, because many other diseases are age-related. In fact, if you look at most of the disease codes in the official classification, I would say about 70% of them are age-related in the sense that their incidence goes up with age. So what we're looking at is a big, big increase in the incidence of disease with age. So we've managed to increase our lifespan, but the problem is that our health span is not keeping up. There's actually an increasing period of ill health at the end of life. And this is actually particularly obvious in women. Women live longer than men in every country in the world, but those extra years aren't healthy years. Women tend to have more ill health at the end of life. So what I can think about this ethically, I mean, it's something one could discuss a great deal, is that in many countries, the major burden of ill health is now falling on older people. And there's really a compulsion to do something about it, not to try to make people live longer. That's not the aim of the kind of research that I and many other people do. It's to try and keep people healthier, to reduce that period of ill health and loss of function at the end of life so that people suffer less before they actually die. So that's the aim of this kind of research. It's called compression of morbidity, trying to make things better at the end of life. And the big thought here is that because the aging process itself is the major risk factor for all these conditions, maybe we could intervene into mechanisms of aging if we could understand what they are. What are the underlying things that are going wrong and causing all these diseases to increase in incidence with age? And could we perhaps intervene directly into those processes and thereby prevent aging-related diseases happening or delay their onset or make them less severe, and also possibly intervene in more than one disease simultaneously if we've got different aspects of the aging process? So for instance, DNA damage and problems with the genetic material generally make a major contribution to multiple forms of cancer. Could we intervene to actually reduce the rate at which DNA damage happens during aging? That's the kind of thought. And that may seem a bit naive and unrealistic, but I think it's really it's the laboratory model organisms that you've been hearing so much about that have really pointed us in this direction. So thinking about budding yeast, Caenorhabditis elegans, the much loved Drosophila, and of course the mouse. You know, can we use these organisms to understand something about the mechanisms of aging that we could perhaps usefully apply to humans? And I think for a long time, people really had a bit of a mental block about this. So they looked at these organisms and said, well, they're completely different from humans. For a start, they live a lot less long. Worms only live about 3 weeks. Flies annoyingly live 3 months. It's a bit too long, but nonetheless, they're very good for aging in other ways. And of course, mice then possibly live for 3 years. But really, these creatures with very different lifespans, very different kinds of physical stresses that they encounter, different kinds of biological enemies. You know, why on earth should they age in the same place, in the same way as each other? I think, you know, the idea that the very powerful evolutionary conservation of mechanisms of genetics, metabolism, and so on might not apply to aging. And the person who's my real hero in all this basically broke that jinx and just did the right experiment. And this was Cynthia Kenyon. So what she did almost, well, over 20 years ago now, was to take Caenorhabditis elegans, simply feed them a mutagen, chemical mutagen, and ask whether she could isolate single gene mutations in the worm that made the worm live a long time. And what she found was that she could. This is the classic paper that I think kicked off a lot of research that continues to this day. And what she found was this mutation in a gene called DAF2 could hugely extend the mean and the maximum lifespan of the worm. You can see this dramatic increase here. And it took ages to figure out what DAF2 was. It wasn't that easy to clone and sequence genes in those days, but eventually it was. And it turned out to be a worm insulin-like growth factor receptor. So much better known in mammals for effect on metabolism in one case and on growth and wound healing in the other. Actually, the presence of the signaling pathway in worms wasn't suspected until this work was done. But here it is, it's an insulin IGF receptor, it's in a worm. And if you produce, it's a partial loss of function rather than a complete null in the receptor, you get this long-lived worm. And this wasn't just an extension of the moribund period at the end of life. These mutant worms were happy and wriggling around and youthful looking long after all the controls were dead. So Cynthia's got some really good films of what these worms look like later in life, and they look extremely healthy. So I think everyone thought, well, you know, that's nice, but it's just worms. But it turned out to be very strongly evolutionarily conserved. This was really where Drosophila came in. So this is DAF2 again, a slightly different experiment from my colleague David Gems, but the same picture, this very long-lived DAF2 mutant. But the insulin IGF pathway came to the light in the flies, largely as a result of the work of Ernst Haffen at that point. And he produced this mutant fly, Chicos, Spanish for small boy. And what you can see is that the homozygotes for this mutant are actually very small. So this is a wild type female and a homozygous mutant female. And you can see that she's also very nicely long-lived, so just like the worm. And Chico is not the insulin IGF receptor, it's the single fly insulin IGF receptor substrate that sits just below the receptor and transduces the insulin IGF signal into the cell. And this dwarfing is irrelevant to this aging story. The heterozygous Chico flies are actually wild type and body size, but they also are long-lived. And this is just two things that the pathway does at different points in the life of the fly. It doesn't have to be dwarf to be long-lived. And it took a lot longer, but eventually this IRS1 null mouse was produced. So this is the exact mouse equivalent of Chico, this insulin receptor substrate. This is a mouse that's null for IRS1. There are actually 4 different insulin receptor substrates in the mouse and in humans. So very typical that there's 1 in the fly, there's 4 in the mammal. But if you knock this one out, you get a nice long-lived mouse. These are data for females. One of the things that is interesting about this nutrient sensing network that this is part of is that they tend to have sex-specific effects. So both Chico and the IRS1 mutant actually extend lifespan a lot more in females than they do in males. But of course, with these mice, it was particularly important to be sure that we'd increased health span rather than just the lifespan of the mice. And this was some of the phenotyping that was done at the time. It's a while ago now. So these two are sister littermates at the age of about 850 days. And this is the, actually, these are labeled backwards. This is the mutant mouse, and this is the control on the right. And you can see that she's got cataracts and also this ulcerative dermatitis that occurs on the head and neck of the mouse. They get less of that. They also get less osteoporosis, and they show much better maintenance of these rather key features. Their glucose homeostasis. So despite being mutant for insulin signaling, actually their beta cells compensate, and they're very insulin sensitive as they age, much more so than the controls. They've got a better immune profile, and they also maintain their motor function into old age better. I mean, we need to do more phenotyping on these animals to be sure that there isn't a downside to these mutants. But so far, one hasn't been found. So that was where things stood until a while ago, when people really started to take an interest in humans, and particularly in looking at whether particular genetic variants in the human orthologs of these genes or the expression levels of the gene products were enriched in people who lived to advanced ages. And these are just some of the papers that have come out on this. And you can see that there really is quite a strong effect here. And these are various either candidate gene approaches, or this at the bottom here was a GWAS. And this GWAS confirmed this rather important finding here. FOXO is a transcription factor, and the single FOXO in the fly and the worm has to be present for these mutants in insulin signaling to extend lifespan. If you take FOXO away, you've got a viable fly, but the insulin mutants no longer make it healthier as they age. So it's a required effector for that. And several repeated studies in humans have shown that particular genetic variants in human FOXO are associated with survival to over 100. And this actually turned up in this GWAS, completely open-ended compilation of the different GWASs that have been done on lifespan worldwide. And it turns up here. So it looks as though at least some of these effects may be important in humans. So the point of all this is, first of all, the importance of nutrient sensing. So what insulin, IGF, and also this TOR that's shown here, which I'll come on to say more about in a minute, what they all do, they're either cellular or systemic systems. So insulin and IGF, of course, circulate and get to most of the cells in the body. And what they do is to sense the current status of the organism. So its nutrient status, is it stressed in some way? Is it infected in some way? And what can it therefore afford to do with its metabolism, its growth, its reproduction, its immune defense, and so on? And hence its importance in aging, because it's weighing up what the defense systems and the anabolic systems of the animal can actually afford to undertake. And as a result of huge amounts of work in various labs around the place, I think we've come to realize that aging does have these characteristic hallmarks. So one of them is this problem with overactivity of the nutrient sensing network during aging. It seems to be set as a result of natural selection in the past at levels that work very well in young people. But they don't work so well as the individual ages. It's too active. And if we tamp down its activity a bit, we can improve health, at least definitely in animals and possibly in humans. And it turns out there are other hallmarks of aging. There are other things that go wrong and that we might potentially be able to twiddle and tweak to improve health later in life. So one I've mentioned already, which is things to do with the genome. So genetic instability of various kinds, including mutations, but also other types of instability, mobilization of transposable elements in the genome, and so on. Attrition of the telomeres at the ends of the chromosomes. particularly in cells that actively divide, alterations to the way the genetic material is packaged and secondarily modified can all have major effects. Various cellular processes, so the way cells handle proteins, known as proteostasis. You know, proteins can aggregate during aging, and there are systems in cells to handle and quality control proteins, make sure that the protein domain's in order. I've talked about this nutrient sensing. Mitochondria go wrong during aging, and mitochondrial density goes down, and also existing mitochondria tend to malfunction. Of course, mitochondria have many metabolic roles, quite apart from their role in respiration. So that can have a very major effect on the cell. There's this process called cellular senescence. So when cells become normally dividing cells undergo genetic damage and other stresses, they can stop dividing, they become resistant to apoptosis, and they produce various cytokines. And in young individuals, these senescent cells are typically produced in the context of wound healing. So they appear, they participate in the tissue remodeling and wound healing, and they're also important for that during development, actually. But then they disappear, they're removed by the immune system. That goes wrong during aging, and they hang around, and the cytokines that they produce can cause widespread tissue damage and also inflammation. So cellular senescence is a major player. Stem cells stop working properly, and cellular communication goes wrong. So the systemic environment goes down. There have been some kind of Frankenstein experiments where people have joined the blood supply of old and young mice, or control old, young mice. And it's clear that there are factors in the blood of young mice that are beneficial and factors in the blood of old mice that are damaging. The systemic environment itself goes down during aging. So these are all potential points of intervention in the aging process. These are called the hallmarks of aging. And I think people are now very interested in how we might intervene and actually use them to prevent things further down the line, actual pathologies from appearing. So this is the kind of picture that people have now, I think. We know that aging is a malleable process. We've seen that with the nutrient sensing network, and with the huge increase in human lifespan. We know that we can keep people and animals healthier for longer. And there are different ways of doing this: genetics, drugs, metabolites. The two that are proving most effective in humans, things that can be done, you know, without any further discoveries, alterations to diet. So amount eaten, the composition of the diet, when it's eaten, are all important. And exercise particularly. It's very clear that exercise at all ages, but particularly in people as they age, can have major effects on various combinations of these hallmarks and combat these things that tend to go wrong on the right during human aging. So we're seeing a picture of different interventions that have weighted effects on these different hallmarks, and that the hallmarks in different combinations contribute to the things that actually happen and go wrong during aging and cause people problems. I think that that's the kind of paradigm that people are working with. And one that I've been particularly interested in for some years now, and where I've actually actively consulted Ross several times and has been incredibly helpful and informative, is possible drugs for targeting these hallmarks of aging to act preventatively, to stop things going wrong. So we're thinking about the kinds of drugs that are used a lot in the context of cardiovascular disease. So for instance, drugs that lower blood pressure, which tends to go up during aging, and drugs that prevent atherosclerosis, that correct the cholesterol and lipid composition of the blood. They're called statins. These are taken by millions of people worldwide purely preventatively. I mean, high blood pressure isn't a disease, it's a risk factor. So the principle of taking a drug to prevent something happening and taking it even over quite a long period is well established, I think. The question is, can we extend that to help people by using drugs to ameliorate these hallmarks of aging and reduce the rate at which they happen? So what I'd like to do for the rest of the time is talk about this drug, which is called rapamycin, or often known by the clinicians as sirolimus. It has these two different names. And at the moment, I think it's the drug that's furthest down the road. It's the one that we know the best and are beginning to understand the best, that seems to be able to ameliorate some of the effects of aging. So what does it do at a purely molecular level? What it does is to inhibit this protein complex here called mTORC1. It reduces its activity. mTORC1 is part of this nutrient sensing network that I was talking about. It's an intracellular protein complex that's particularly noted for its ability to sense amino acids, but it senses an awful lot of other things too, which are shown here by the input arrows at the top. And it also has a massive number of outputs. All these different cellular processes are affected by the activity level of mTORC1. So it's an absolutely key central cellular regulator of sensing what's going on and deciding what to do about it in terms of cellular processes. And what rapamycin does is to inhibit its activity by disrupting its interaction with another regulatory protein. So that's what we know it does in a biochemical sense. Rapamycin is a natural drug. It was isolated originally from bacteria on Rapa Nui, Easter Island. There was an expedition that went to look for soil microbes that could inhibit cell growth and proliferation. It was basically a search for anti-cancer drugs. And rapamycin was the one that turned up. That's hence its name, rapamycin Rapa Nui, because of where it came from. And probably because it is a natural product and it's evolved in the context of wars between different microorganisms in the soil. They're trying to inhibit each other when they're competing. It's an extraordinarily effective drug with very evolutionarily conserved effect on this TORC complex, which is what it's evolved to actually work against. So what does rapamycin do? Well, for a start, it extends lifespan in flies. So this is Ivana Bedov, who now has her own lab at the UCL Cancer Institute. She was a postdoc in the lab for a while. And what she showed was here that rapamycin can extend fly lifespan. It's a somewhat modest increase, but it's extraordinarily repeatable. And it happens in the context of a long-lived, very healthy wild-type strain of flies. So you can see the more or less dose-dependent increase here. And I'll come back to this, but what you can do with drugs, Drosophila is very powerful because, of course, you can overexpress, knock out, underexpress, modify genes, and you can look at how they interact with the effect of the drug treatment to try to figure out what kinds of processes might be being affected. And in this study, she actually pinpointed that something called the S6 kinase, but particularly this lot that are involved in a process called autophagy, were important. I'll come back to that in a minute. So it's nice that it extends lifespan in flies, but I think what's more important and what really struck people was this, which is that it very reliably and substantially increases lifespan in mice. So this is from the American National Institute of Aging study. They have a competition each year for drugs to put through their study, and they test them in 3 different centers in the United States. And you can see this extraordinary increase in lifespan. So dose dependent, greater in females than in males. I wouldn't want you to go away with the idea that it works better in females. What actually is presented here is the concentration of the drug in the food. It's fed orally to the mice. If you look at the concentration in the plasma, actually for an amount of rapamycin in the food, the plasma concentration is lower in males. They detoxify it more rapidly than the females do. And actually dose for dose in the plasma, the response in terms of lifespan looks rather similar. And these mice have been, of course, extremely thoroughly phenotyped. And the drug really does seem to reduce both the incidence of cancer, as you'd expect from an anti-cancer chemotherapeutic, but also many other aspects of the aging process. It really does look like an anti-aging drug. And very importantly, this work and other work with rapamycin was picked up by somebody working in the Novartis drug company called Joan Manick. And she became interested in the possibility of actually doing clinical trials with rapamycin in humans. And the context that she picked was actually very interesting in the context of COVID. It was response to immunization against flu, because old people are both more vulnerable to flu. They're more likely to have a bad go with it if they get it. But also, if they're immunized against flu, their response to the immunization is weaker. They give a less good immune response than younger people. The same is actually true in mice, and this was where the initial experiments that Joan picked up on were done. But what she did was this study, which was to look at what happened if she pre-treated humans, actually not with rapamycin itself, but with another drug combination, everolimus and a kinase inhibitor, that effectively do the same thing. The point of using the combination was that half of it's a proprietary Novartis thing, and the other thing is that the combination is different from what's been used before. We were talking about patenting in the previous talk. They needed something that they could patent if it worked. So that's why they used this particular drug combination. And what they found was that if they pre-treated people with rapamycin versus controls, of course, then waited, let them clear the drug, immunized them, and looked at what happened. First of all, the immune response to the immunization was greater with rapamycin pre-treatment. And also the number of respiratory infections in the ensuing winter was down. So it really did seem to boost the immune response of these older people to immunization. And actually, they subsequently went on to show that actually, if they left out the immunization and just gave them the rapamycin, that itself protected them against flu in the ensuing winter. So it's a very interesting drug. It's one that looks as though, you know, it really may be going forward to humans. So what about flies, which is the point here? How can we use Drosophila to understand how rapamycin is ameliorating the effects of aging? Well, as I've said, it's a complicated, picture. How do we get through the thicket? And to tell you the answer, and then I'll tell you, or at least what we think is the answer, and how we got to it, we're looking over here at this process called autophagy. So rapamycin inhibits TORC1, and that activates autophagy, because normally TORC1 in the absence of inhibition inhibits autophagy. So what is autophagy and why would TORC1 inhibit it? Because TORC1 is a let's go ahead and be anabolic, everything's fine protein complex. So what autophagy does is to actually deal with damaged cellular components, so macromolecules, but also organelles and other things that become damaged in the cell. And what happens is this isolation membrane forms first, and you then get this proto-autophagosome, and then eventually the autophagosome. And what it does is to accumulate the damaged components that need degrading. And then at a certain point, it fuses with a lysosome where they're degraded and then ejected from the lysosome. And this happens in two contexts, really. There's a basal level of autophagy, just a routine, dustbin emptying process that goes on in cells. But also, if there's starvation, then autophagy is activated as a means of producing key metabolites. So there's a basal level, and the process can also be activated, and it normally is under starvation conditions. So that's why TORC1 inhibits autophagy, because everything's fine, so nothing more is needed than the basal level. It's only when starvation happens and TORC1 is inhibited, this time by nutrient missing, that we see the activation of autophagy. So how did we get to this conclusion, and what was the power of the fly, if you like, in doing that? I think the first thing that gave us a bit of a hint and helped to find a way in is that there's a big sex difference in the response to rapamycin in flies, much more so than in mice, which is that males don't respond at all. So this is a survival curve for males that were treated in rapamycin in the dark blue, or for females that were treated in the dark red. And what you can see is the extension of lifespan in the females that you've seen before from Ivana's work. But if you do that in males, you don't see the extension. This is Jenny who did these experiments. She now has her own lab at Edinburgh University, where she's working on fly immunity, actually. So there's a sex difference. And with flies, that's rather a big hint about what might be going on, because there's a very big difference in the way that the gut ages in male and female flies. So this is shown here, females at the top, males at the bottom. So these are now obviously microscopy pictures of the gut. This is Rasil, which lets you see the membranes of the cells in green. So we've got flies getting older towards the right. And in the intestine of the fly, or this region of it, it's very like a mammalian small intestine. You've got these columnar cells sitting on a basement membrane with the other end facing the lumen of the gut. So you can look down on top, as is shown here with this male, you get this nice honeycomb arrangement, or you can take a section through the gut, as is shown here, and you've got these nice columnar cells lined up. Here's the basement membrane, sorry. Here are the nuclei and dapi, nice orderly columnar epithelium. And as the male gets older, nothing happens. I mean, basically males die with their guts really looking very nice and intact. Whereas with females, the picture is very different. Even by the time they're 3 weeks old, things are starting to get a bit crazy. You can see that these nuclei are out of alignment. We're starting to see missing cells. A bit later on, we start to see these clusters of cells near the basement membrane. They're known as tumors in the fly gut. They're not really, they're just misdifferentiated enteroblasts. So in the gut, stem cells are present, and they divide to give rise to the three main differentiated cell types in the gut, which are the big absorptive enterocytes, the enteroendocrine cells, and of course the stem cells themselves. And what you find is that as the gut ages, this differentiation doesn't happen properly. The stem cells get kind of stuck at the enteroblast stage, and you get these little clusters of cells, so that the epithelium starts to actually come away from the basement membrane. You get these wound healing rosettes, and by the time the female's old, the whole thing's a complete horrible mess. It's amazing really that the gut works at all from its appearance. So we've got this sex difference in the two things. And one of the really, really useful things about flies, and you've heard this touched on in the previous talk, is that the sex determination is cell autonomous. So each cell in the fly decides what sex it's going to be in accordance with its own genotype. There are no circulating sex hormones. And so what that means is that there's a sex determination cascade. The genetics of that are very well worked out by a lot of very clever people. And it means you can manipulate it experimentally. So what you can do is to give a male fly a female tissue. You can transdetermine it using a genetic trick, or the other way around. So what Jenny did very cleverly was to say, right, I'm going to give females male enterocytes, these big absorptive cells, and I'm going to do the opposite. I'm going to give females male enterocytes. And I'm going to ask whether the sex of the enterocytes in the gut determines the response to rapamycin treatment. And she found that it did. So this is shown here. We've got males. Don't worry about this MEX system, which is just the genetic trick that's used to transdetermine the sex of these cells. And she used a number of assays to show that she had indeed transdetermined correctly. So that the experiment worked at the genetic level. What she found when she did it was that if she gives females male enterocytes, which is the blue, they no longer respond to rapamycin. Whereas if she gives the males females enterocytes, they now respond to rapamycin with an increased lifespan. So what this transdetermination has let us do is to figure out what tissue is important, at least in the initial response to the drug, to the rapamycin. So I think flies are very useful in that way, the fact that you can do that. So that meant that we could focus our attention on the enterocytes and what was actually going on in them. So we did that. And we were very interested in autophagy simply because it is one of the main outputs of TORC1, which we know that rapamycin inhibits. And autophagy is a very annoyingly difficult process to measure. What you ideally want to look at is the efflux of these damaged components through the pathway. You'd like to look directly at what's happening at the flux level, but that's extremely difficult. So we used a number of indirect methods. We can look at the expression of components of the autophagy pathway, or we can use this rather blunderbuss approach here, which is to look at autolysosomes. So there's a drug called isotracker, which enters acidic vesicles and which therefore enters the autolysosomes that are present during autophagy. You would only ever use it in conjunction with other methods, which we did. But it's nice because it's visual. And what you can see is illustrated here. The quantification of this is over on the right, but the picture tells the story. So basically, if we give females rapamycin, they start with a very low level of basal autophagy up in this square, but with rapamycin, up it comes. With the males, oddly enough, they have a high level of autophagy in the gut, just at basal level. For some reason, males have higher autophagy activity than females do in their guts. We don't know why that is or what its functional significance is. But be that as it may, when we give them rapamycin, it doesn't increase. This is shown over here. There's no significant increase in response to the drug. And actually, if we trans-determine the guts, this again, making them female in the male and male in the female for the enterocytes, what we find is this phenotype goes with it. So whether or not you have a high or a low basal autophagy level, and whether you respond to rapamycin, depends upon whether your enterocytes are determined as male or as female. So that seems to be what's going on here. So that was one important line of evidence that suggested to us what was going on. And another that gave us an interesting handle on it, and that I also think has some medical implications, was discovered by Paula. So Paula's a PhD student and then postdoc in the lab in Cologne. She's now actually a postdoc in Lausanne, working in a lab there. And what she found was something rather extraordinary about rapamycin, which turns out also to be true in mice, which is that if you let your flies come out as adults and just feed them with the drug for a couple of weeks, days 1 to 15, after they've popped out of the pupa, and then take the drug away, you can look at what's going on at the signaling level. And what you find is that TORC1 activity recovers completely. So they clear the drug, we can show that it's not there. We can show that TORC1 activity recovers completely to normal wild type levels. So we've got this fly with normal TORC activity, but that has had rapamycin in the past. If we just wait and look what happens when they die, this is what happens. So the flies with the history of rapamycin treatment all that time ago live just as long as flies that are chronically treated with the drug throughout their lives. So they've got this astonishing memory of having been treated with the drug in the past. I actually didn't believe this result the first time she did it. But I mean, it's definitely true, and it looks as though something very similar is happening in mammals as well. So of course we became very interested in what was going on here. And one of the things we did, given that we already knew about the sex difference in gut pathology and all of that, was to have a look at what was going on in the gut. And there are various ways in which you can assess the function of the gut. The histology I've already shown you with the tumors and so on. And what she found was that if she gave them, initially she gave them the drug for almost a month, she shortened that subsequently. If you look 20 days later at day 50, flies that are either still on rapamycin and have been chronically treated with it, the orange, or were treated in the past, days 1 to 30, or that never saw the drug, then what you find is that at day 50, tumors are up in your controls that are never treated, but they're just as low in the flies that were past treated as ones that are chronically treated. So these clusters of improperly differentiated enteroblasts are just as big a problem, or rescued just as well by past treatment as by chronic treatment. You can also make various, in the fly, rather nice functional measures of how the gut's doing, because you can measure gut leakage. So the way that's done is simply by feeding the flies a harmless blue food dye in their food. And in a normal fly with a healthy gut, the dye stays in the gut. You can see it here. But if the gut's leaky, you get the smurf, a blue fly. And again, if she does exactly the analogous experiment to here, this is the smurfing rate in the controls. It's way down with chronic rapamycin treatment, but it's down just as much if they've seen the drug in the past. So somehow the health of the gut at old age in these flies is being rescued by much earlier treatment with the drug. And this all seems to boil down to autophagy, because again, if she does early rapamycin treatment, you get just as great a long-term increase in lysosome numbers in the gut as you do with chronic treatment. So again, this is quantified here. So with chronic rapamycin treatment, you see a bigger increase, a big increase in autolysosomes. This is this comparison here, and the red lit up bit is showing us the autolysosomes with the DAPI nuclei. If they were treated day 1 to 15, and then she looks at 12 days later, again, the autophagy stays up. And the interesting thing here is, again, one of the huge advantages of flies is that you can manipulate autophagy directly, genetically. So there are a lot of different autophagy genes. They're all called ATG something. And one of them, ATG1, which is the equivalent of mammalian Becklin, if you overexpress it, you can induce autophagy in the fly, and you induce it in the tissue where you want it, depending on what GAL4 driver you use. And the other very important thing in flies is that there are these wonderful inducible systems for gene expression. So you can have a modified GAL4 driver that isn't merely tissue specific, so you can control its expression spatially in terms of the type of cells it's expressed in. You can also control it temporally, because the GAL4 only binds to the upstream activating sequence and activates the whatever it is that you're expressing. It could be an RNAi, it could be a positive gene, but whatever it is, it only activated in the presence of a steroid hormone inducer. So what that means for our purposes is that you can rear the flies through to adulthood without any developmental issues at all. There's no gene misexpression during the developmental period. But then when they come out as adults, you can feed them this steroid in the food and induce the expression of whatever you want. And what we did, of course, was to do that with autophagy using HG1. And the results of doing that are shown here. So there's chronic treatment on the left and early treatment, days 1 to 15 on the right. And this is in enterocytes. So this 5966 gene switch is the name of the system where you can temporarily control the expression of the transgene. And we just controlled it to the adult. And what we're doing is overexpressing HG1, which is one of the ATGs, where if it's manipulated, you actually get a response in the autophagy process. And what you can see here is, first of all, chronic rapamycin treatment. So the dark black line as opposed to the dotted, these are females, of course, and we see the nice increase. If Paola chronically overexpresses ATG1, so she feeds the flies the steroid hormone for the whole of their adult life, then she gets an exactly equivalent increase in lifespan to what she sees with rapamycin. And if she puts the two together, then she doesn't see any further increase. So what that argues is that these two interventions are acting through the same pathway. They're epistatic to each other. Either alone is sufficient to get the full effect of putting the two together. So I think this suggests rather strongly that autophagy is the really important process here. This is what happens with the equivalent memory experiment, and she gets exactly the same result. So what she does here is to either give the flies rapamycin on days 1 to 15 and then take it away, gives them the steroid hormone inducer days 1 to 15, so induces autophagy, but just for the first 2 weeks of adulthood, and then takes the inducer away, or puts the two together. And she gets the same result, which is that she gets the increase in lifespan as a result of the early rapamycin treatment. As we've seen, the flies have a memory of it, but they have an exactly equivalent memory of having had autophagy induced in this period, and she gets the same extension of lifespan. And what we're actually seeing here is that autophagy, is, at least in the context of these fly enterocytes, is a rather peculiar process. It actually induces itself. So if we turn on autophagy in these cells early in adulthood, it stays up for the rest of their lives. And we're in the process at the moment of trying to figure out why that is. But something very similar seems to be going on in mouse guts. I mean, it really does seem to be conserved in mammals. And I think this could be a very useful way in to using induction of autophagy to improve health during aging.
Linda, if you can wrap this up, it'd be great.
There you go.
Like that.
So to summarize, what we've got here is a malleable process that can be ameliorated by mutations in single genes. Nutrient sensing networks are important. Drugs can be used, including rapamycin. I think we might be looking here at a quite broad spectrum preventative medicine, and DrosAfrica is just a hero for studying the processes of aging. So that's it.
Fantastic. Thanks, Linda. That was amazing, as always. So I'm going to keep the questions tight, and then we'll put them up on Slack to keep us on time. So I think there are a number of questions that we're sort of getting to a similar issue, which is, what is the future of these rapalogs for aging? And more generally, what is the challenge going to be to identifying a true, maybe not improved lifespan, although that would be great, but health span? What would be the challenge for health span drugs? Like getting them through clinical trials?
I think the obvious one is side effects. So we know that rapamycin and other drugs like it, when they're used at clinical doses, so rapamycin is used to prevent tissue rejection after transplant cancer chemotherapeutic. Those are the two main ones. And at the doses it's used clinically, it does have some quite nasty side effects. These doses that are being used experimentally are much lower, much lower. So I think it's going to be crucial that any repurposed drug can be used at low enough doses that the side effects are minimal. I think the other things, clinical trials, I think it's going to be really important to do what Joan did, which is to find somebody targeted problem that happens during aging and work on that, and then gradually build up to the wider effects of the drug. I mean, the metformin trial, the TAME trial, has now been FDA approved. So this is a trial in the States. They haven't been able to finance it yet, but it's got FDA approval, which has got aging as the outcome for the clinical trial. I mean, that's a first. But that means looking at multiple diseases. So to get at the statistical power, you're going to need a very big sample of people. So it's expensive. That's why I think that I suspect there will never be drug development for aging. I think it's just too impossibly expensive. It's going to be repurposing.
Yeah, I would probably agree with that. I'm just going to have one more, which is Marianne. I see you have your hand up. Does that mean you want to ask a question? Sell it in?
Yes, Prof. Ross. Can I go ahead?
Yeah, go ahead.
Okay, I ordinarily will have just typed in these questions, but I feel the words are so much, and I'm feeling lazy to type. So I'll just say it. The first one is when Prof. Linda was presenting, she said basal autolysosome activity was higher in male flies than in female fly, and she couldn't explain why. So I am kind of trying to give possible reason why this is so. From yesterday's presentation, we're made to understand that the females feed more around the time of mating. So I think if this was not put into consideration when taking the basal lysosome activity, it could be a contributing factor. Perhaps the autolysosome activity was was checked when these females were starved. And since starvation, sorry, when these females were well fed and they were looking healthy. And since autophagy is inhibited only in starvation, this could explain why it was low in the females. I don't know if they put that into consideration before taking note of
Yeah,
the
no,
basal.
I think it's going to be something like that. I completely agree with you. One of the obvious things about flies is, you know, they're a bit different from mammals in that the sex difference is much, much bigger. So females are basically machines for pumping out eggs. So they have to eat huge amounts of protein. They're very anabolic because they lay a lot of eggs. Whereas males basically just need to tear around finding females. They don't have that problem. So they eat a lot less than females do. So the female part is just under much more, you know, it's much more heavily used just to get nutrients into the fly. And I suspect that's going to have a major role in that difference. So I quite agree with you.
All right. And then the second one is considering the use of rapamycin in human trial. I think, anyway, this from a very unserious point of view, I think it shouldn't be much of a problem because everybody wants to live longer. And I'm sure a lot of people would like to volunteer to undergo this kind of clinical trial. And who knows?
Do you really think everyone wants to live longer? I think people want not to get sick, which is a bit different.
Yes. And from the presentation, it's already obvious that rapamycin is increasing health span in the fly. So if people know that there's a high probability of them having healthier life, they might really want to key into the clinical trial. I, as a person, will key into the clinical trial. And then eventually, if it passes, then maybe we'll be thinking of using rapamycin supplements to increase healthy life. That's just on a lighter
Yeah, we
note.
might do, but you saw all the things that you saw all the different things that top one does. It may be that we should be targeting the autophagy process more directly with a different kind of drug. I don't think it's clear what the best way into that one is yet.
Sure, that, and also senescent cells, and so it's going to be an interesting time to see what to dig out. Okay, Linda, I'm going to end this, and we're going to move to our final talk. So, Linda,
thanks, everyone.
Excuse me.
I was just saying thanks to everyone. You know,
you're not truly done. We're going to put the questions, and there are a lot of them over on Slack. So if you have a moment to check out Slack, and if you all want to put some of your questions on there, we'll also try to push some on there. I actually have quite a list, which is why I just had to finish. So thank you, Linda.
Cool.
So Mohammed Salem, are you here? Mohammed? No, I don't see him listed here. Okay, I didn't expect that. That's all right. So I think while I give Mohammed a few more minutes to see if he's here, perhaps I will. Linda, since you foolishly did not sign off, I'm actually going to ask you a few more questions. You really are going to regret not signing off. And then Mohammed, if you are here, just let me know. So there's been a lot of, so one question by Audubon Bismarck Takura, also a similar question by Mariam Celadine. With the advent of genomic editing, what about the possibility of editing genes with aging people, especially because you show that you can target them not to the whole body, but maybe you could find critical cells. Gut would be a great place to start. One could imagine like liver, maybe brain. What about the potential to target those? And of course, there are a lot of ethical issues that come with that as well.
Yeah, I would have started with the ethics. So there's just been a big National Academy of Sciences Royal Society WHO report on gene targeting in humans. And I mean, to cut to the chase, I think the conclusion is that it would not be ethically justifiable for the foreseeable future. We just don't know enough I mean, quite apart from any other ethical consideration, the off-target effects are still just much too dangerous. It really is not a justifiable intervention for anything at the moment.
Yeah. Yeah, I guess there are some disease issues one could explore, but yeah, basically. And what is the underlying, Wassem Meshrif wanted to know, what is the underlying mechanisms in the differences in guts between males and females? And also, do you see these sorts of differences in tissues in humans?
Oh, no, that's really interesting. So the fly, as I say, I think the gut is just set in completely different ways in the two sexes because it has to function so differently in them, because of females needing to absorb so much food, and also to deal immunologically with the problems that come in with eating a lot of food, because of course, you know, fly food is out there and associated with microorganisms and so on. So I think a lot of the gut difference will boil down to that fundamental difference in biology between the sexes. What I discovered through talking to Irene Miguel Aliaga, a very well-known fly biologist, that in mammals, a quite extraordinary thing happens in females during pregnancy and particularly during lactation, which is that the small intestine hugely increases in length. So there's a massive cell proliferation and growth in the gut in females under those circumstances. So the gut is first of all set up to do that, but also afterwards, and associated with parity, the pathologies that develop in the aging gut are different in men and women, and in male and female mice. So it's not same-sex differences in flies, but there certainly are sex differences in the gut. And more generally, I'd say that the gut's been a really largely ignored tissue during aging. But of course, it's the first point of encounter with all sorts of things from the outside world. And it wouldn't be surprising if it played quite a major role. And in both mice and humans, as people age, and as mice age, the gut becomes leaky. I mean, that problem is present there as well. So I think there are similarities. I also think there's a huge amount we don't know.
Sure.
There's a lot to do.
Sure. And I know you've worked on some other pathways. Did you just want to say a few words about some of the other pathways that you've been exploring?
Well, the insulin IGF signaling has thrown up a lot of different things. I think one of the main things that's come out, I mean, in some ways it's rather obvious, but the different tissues have to combine to increase health during aging. And in the context of reduced insulin signaling, it's becoming very clear that both the molecular responses, actually, depending on what players are present in different tissues, but also the outcomes of those responses, you know, what aspect of the aging process is thereby tackled, is very tissue specific and even cell type specific. So there are particular things that happen in the gut, what happens in muscle and fat body are different. So proteostasis turns out to be really important in muscle, for instance. And also mitochondrial density is very important there. So there are different tissue specific things that are all ameliorated by taking down this one hallmark, the activity of the nutrient sensing network. So there are quite complicated interactions between different aspects of the aging process in different tissues. I think that's come out. And there's a whole little molecular world there of, you know, microRNAs, circular RNAs, weird RNAs that are part of the effector process as well. So, I mean, an almost bewildering number of potential targets in different tissues. And of course, clever drug people like you are managing to make drugs themselves more and more tissue specific. And I think that's going to be very important, you know, being able to target particular drugs to particular cell types. I think that's going to be key here.
Well, it's interesting you mentioned that because if I may, so in diabetes, you probably know this, but one of the difficulties with diabetes drugs is that different cells, some of them actually take in glucose passively, for example, and then you crank up the sensitivity to glucose to drive it into cells that are insulin resistant, and you end up blasting some of these other cells like muscle cells and neurons with glucose. And so over a long period of time, these insulin sensitizers end up causing a lot of problems themselves because you treat all cells.
I didn't know that. That's really interesting.
Yeah, so I wonder in aging, I wonder if in aging, as you begin to, as you say, if you can fine tune it, one could imagine that some cells are going to take some of the agents more passively than others, are going to require larger doses of, rapamycin than others, and so on and so forth. I was imagining, for example, where you showed that giving rapamycin early was sufficient. One possibility, of course, which I'm sure you've thought about, is if you give it at a reasonable dose early and then you tail the dose off later, you're not fighting the toxicity of rapamycin later, which it may become more toxic. And so you have an additive of extending more lifespan, but also toxic, and it sort of adds up into about the same.
Yeah.
So this sort of refinement cell by cell is,
Yeah,
I think it's interesting.
it is. It is.
But for now, I'm going to wish everybody goodbye. Thank you very much for participating. Many of you have asked about posting these talks. No worries. We are recording the talks, and probably what I'm going to do, instead of making you grind through the whole thing, is I'll try to chop them up into the individual speakers, and then we will put them somewhere where you can get hold of them, go back over them. They're actually also transcripts being automatically generated. I don't know how accurate those are. And I will post those as well. So we will do everything we can to allow you to go back through and sort of dig into some of the talks that you found particularly interesting. So much appreciated. And we have one more session tomorrow. Much appreciation and love to everybody. And I will see you tomorrow. Be good.